Organizing committee for inviting me here to present the work we are developing to fight against this important plant pathogen. So this is the outline of my talk. I will present you who is the Xylella fastidiosa. The most important part is how Europe reacted to fight against this plant pathogen. Then I will present the BCIL project. It's a Horizon Europe consortium that I'm leading from the Institute of Sustainable Agriculture in Córdoba. And then I will present some of the main results that we are taking and the activities to fight against this bacterium, mainly focusing on preventive measures. So this plant pathogenic bacterium is a gram-negative bacterium. It's fastidious, so the name comes from the difficulty of growing the bacteria in vitro and working with it in the laboratory. So the bacteria has a dual cycle life. In fact, the plants only inhabit within the xylem vessels, but also is transmitted by different species of vectors, mainly from the family Cicadelidae, Cercopidae and Frophoridae, so it has a dual lifestyle. Most importantly, it has a very wide host range. It can infect more than 700 host plants, but only in a few of them, not many, not the 700, cause very important diseases. And it's able to establish in different climatic conditions. And also the most important thing is that when it is established in a territory, it's almost impossible to eliminate. So another important fact is that Xylella fastidiosa is not only one single thing. It's composed of different subspecies and different genetic groups. Up to the 90 different sequence types has been described in the world. And it's very important to identify which species we have in each country or in each area, because if we know the subspecies, we know which is the host range that the plants that can be affected. And also, it has been recently discovered that the different subspecies can recombine between themselves and generate new variants that can infect new host plants. Xylella not only causes diseases in olive, but it causes diseases in grapevine, citrus, peaches, also different symptoms of leaf scorch, very general symptoms, not very specific, in different host plants, crops, but also forest trees and fruit plants. So the bacteria was first discovered in Italy, in the hill of the Butte in Apulia region in 2013. And the symptoms start with a pale green color of the leaves that start very fast developing necrosis through the different branches, and finally collapsing the entire tree of the very susceptible varieties. So this happens through hundreds and hundreds of hectares and affects thousands of trees in the region. As I told you, the symptoms started in this region, in Gallipoli, and at the time that the bacteria was identified as associated to this decline, already 8,000 hectares were already affected. In a few years, in 5 years, the infected zone spread over 100 kilometers, and at that time, more than 25 million of olives were already infected by the bacterium. Currently, the situation is that the bacterium has been identified in the north near the city of Bari. And you can see here the red triangles represent infected trees, and each single triangle in green, it means a tree that has been analyzed by molecular tests to identify if the tree is infected or not. You can imagine how many thousands and thousands of analyses are being performed every year in the region. So just to see how fast the epidemic develops, you have here a centenary olive tree in 2014. You see small branches that are starting to desiccate, and 2 years later, you see the tree almost collapses. The person at the bottom is myself, just taking pictures. This tree is completely gone nowadays, so there is no tree. The problem is that each of these singular trees has a name, so they belong to the history of the region, and the grandfather of the farmers know that this tree was present when they were still childs. And here you see the typical picture from internet in which you have in 2013, 3 years later, all the trees completely collapsed. In 2021, completely the farm was removed. And I have to tell you that nowadays there is a new olive plantation of some varieties that are shown to have some tolerance to the disease. So there is still hope in the region. So what happened after the detection of Italy is that it was mandatory to all the state members to do surveys. And we found that Xylella fastidiosa was present with us for many, many years, and we have no idea at all about that. So the last detection has happened in Extremadura region. It's caused by a different subspecies, not affecting olive trees, but it can cause many problems in grapevines. So hopefully we can eradicate before it spreads to the grapevine plantations. Another important factor is people thought that Xylella was jumping from Italy to other regions in Europe, but it was not the case. So currently, we have identified 4 subspecies of the bacterium and more than 12 different genetic groups. So Europe has been importing plants from all over the world where the bacteria was native. It was established in the territory, and until we have not searched for the bacterium, we have not found it. Of course, when you have some problems, because the bacterium is very difficult to isolate, people doing traditional isolation techniques in the lab could never isolate Xylella. Only till we have employed molecular detection approaches is when we have noticed that the bacteria was with us. And we have very recently new detections, so as I told you, in Extremadura, in Puglia region, not affecting olive, but also affecting grapevines, and also in Portugal. So Xylella was ranked, it was already a quarantine pest for Europe, but it was ranked the first priority pest among 20 of the one analyzed by the GRC. And this was taking into account different indicators, economic, social, and environmental. So in 25 of them, in 13, Xylella was ranked first. So you can imagine the important losses that this bacteria can cause. So in a scenario of completely full spread, it was estimated that 5.5 billion euros per year could be lost. Mainly in olive trees, but also in citrus, almonds, and grapes. Also, the social cost of Xylella full spread will be enormous because it can affect many jobs involved, not only directly farmers, but all the secondary jobs related mainly to olive. Also, different agricultural products covered by quality labels could be affected. Different plant species that are present in UNESCO World Heritage places. Also, the environmental cost will be enormous because of insecticide application for stopping the vector, and also because many habitats of different species could be also affected. We saw in the previous session how important is olive crop for maintaining biodiversity. So it's not only the cost of losing the olives, it's the cost of losing the landscape and also the biodiversity surrounding the olive crops. So the major efforts are put in Europe for early detection and for surveillance. So if we evaluate how much could be the cost production loss as compared to how much costs the surveillance, you can imagine that it's worth to put money in doing surveillance. So this is the current situation in Europe. You see in red the areas under eradication. That means that if you find a positive plant, 50 meters around the plant, all the host species have to be eliminated. It doesn't matter if it's infected or not. All of them have to go, okay? And the blue ones are areas under containment. That means that they have assumed that it's going to be very, very difficult to eradicate Xylella. So different strategies have to be implemented to try to suppress or to diminish the effect of Xylella. So you see here in Spain, we have Alicante, we have Extremadura, 3 islands in the Balearic Islands. So how Europe reacted to this problem? So You see here the different detections in the 4 countries affected by the bacterium, how EFSA provides support for scientific advice, and in parallel how the European legislation was producing new decisions. So I have to tell you that Xylella is the most regulated plant pathogen all over the world. This happened because many different research projects started, were financed by the European Union, for regional governments, for the interprofessional of olive oil. As the science was providing answers to the problems, the legislation was being modified. So this is a really important fact that the science really provides support for policymakers. And currently, EFSA has been requested to update the quantitative pest analysis on Xylella fastidiosa. As a member of the Plant Health Panel, I am in charge of organizing this scientific opinion. So we are really working hard to update all the information concerning this plant pathogen. So in a recent review, we focus on what were the real challenges that we have to understand and manage this bacterium. And today I'm going to give you some ideas of how climate change, because this session is also related to climate change, what is the uncertainty of the epidemics of this bacterium, what can we do for early detection of this pathogen that, as I told you, is the best way to control the bacterium. If we can avoid the entrance or the establishment of the pathogen, it's much better than when it is established, try to do something. And also I will talk to you about the big collaborative efforts, research efforts to combat this bacterium. So now I'm going to present, as an example, the BCIL project. So as I told you, there were many research projects that were developed since 2013 to fight against this bacterium. So in 2021, we have the EFSA conference, the workshop on Xylella fastidiosa. And at that time, different stakeholders still have some research needs that were not answered at that time. So the farmers, nurserymen, managers, risk managers, and legislators. So at that time, we decided to apply for a new project, BCIL project, that means beyond Xylella. So we want to, okay, now the epidemic, the crisis has passed. We know that we need to start learning how to live with the bacterium. It's time to think forward. And for that, we developed different BCIL-specific objectives adapted to each of the needs of the different stakeholders. I'm not going to read all of them, but I'm going to summarize what is the consortium. The consortium is quite big. We are 31 partners, 28 beneficiaries from the European Union, from 14 different countries, so include countries where the bacterium is native in America. So we needed to take benefit of the experience that they have forever and many years working with the bacterium. And also because in Europe, since it is a quarantine pathogen, you need very specialized facilities, biosecurity containment 2+ facilities to work with the bacterium, so it's not easy. And of course, we have most of the countries in Europe that are affected by the bacterium. This is the structure of the project. So we have to move from the concept of olive being a priority, because it was, of course, our first priority. But now we have problems in almonds in Spain, very important in Alicante and in the Balearic Islands. We have problems in grapevines in Spain and in Italy. So we need to think more global. So, and also we have to provide impact and solutions for the European Commission. The first one is to enhance capacities to prevent, monitor, and find adequate responses for this bacterium. So, of course, we are exploring new knowledge on the biology of the bacterium, how climate change can affect the bacterium, developing thermotherapy treatments for the nursery industry, because it is also very affected by this bacterium. And also concerning early detection, developing new molecular approaches for the early detection of the bacterium, early phenotyping. So we need to take advantage of the hyperspectral cameras to detect the bacterium before symptoms appear. We are using dogs, and they are very good to detect plants that are infected by the bacterium. Also remote sensing to explore all the territory and find hotspot of infected trees. Concerning the impact tool, that is to enhance the capacity to control the bacterium once it's already established in a place. So we need to focus in the disease triangle. So developing tools within the plant, so searching for varieties that are more tolerant or are resistant to the bacterium. Focusing on the vector, we need to develop biocontrol tools, either predators or entomopathogenic fungi that can kill the vector selectively. And focusing on the bacteria, working on synthetic microbial communities, biocontrol agents, bacteriophages, antimicrobials, either antimicrobial peptides, plant extracts, etc. And of course, as I told you, the most important part is not only do research that we really love, but to provide information to the relevant European plant health policy. So we need to give them the answer how to evolve with the legislation. If you are interested, I invite you to visit the BCIL webpage. There you have a lot of information about the different deliverables of the project. We have a newsletter, we have open access documentation, the publications. We are very active in social networks, and also we develop different seminar series every 2 weeks, more or less, with different research groups participating, not only those that participate in BCIL project, but we are open to all the community interested in Xylella, and we are over 600 people already engaged in the seminar series. So just to present a few main results and activities concerning the project. As I told you, one of the main focus is to detect the bacteria before it enters in a territory, or once it enters before it is established. So what tools do we have? So one of the things is to develop pest risk assessment models under current and climate change scenarios. The second one is to develop remote sensing or proximal sensors that can detect the bacteria before it's visible by the naked eye. As a plant pathologist, you can see a plant that is completely looking healthy, but Xylella has a very long infection process before the symptoms appear. So what you cannot see with your eye, some specific devices can see for you. And then, of course, the new molecular diagnostics protocols to detect the bacterium. So concerning risk model analysis, this has been mainly performed by my colleague, Dr. Navas-Cortez. So if we know where the Xylella has been established worldwide, we have a predictor, climatic data, so using different model estimation, species distribution models and other different models, you can estimate the potential distribution under current and also future climate change scenarios. So what we saw is that the suitable areas for establishment of Xylella in Europe are characterized by warm temperatures during summer, dry period, mild winter, and well-defined rainy season. So this is what is expected, the Xylella distribution in general, but it's very important that not all the subspecies could be well established in all the areas. So that is the reason I told you once an outbreak is formed, it's essential to identify very soon which subspecies is present in the area. And what we know is that Europe has climatic conditions that will allow the establishment of the bacterium in many, many different regions, of course being the southern regions most at risk. And then taking into account the different climatic data, the time periods, and the different representative concentration pathways and the different models. So my colleagues also estimated what could predict in a global expansion scenario for Xylella fastidiosa. So we have very bad news, so it is expected that the suitable area for establishment will increase in all the different climatic scenarios, but the severity of the suitability will be a little bit different. But in general, globally, it will increase. We are also developing specific regional models because if we can combine also the land cover and all the different crop maps with the climatic data, these models can be improved. Also, using a collection of a huge number of strains from different subspecies, different crops, and different regions. This student is already a postdoc. He finished recently his PhD. Estimated what was the effect of minimum and maximum temperatures for Xylella fastidiosa. What he saw is that the temperatures below 10 degrees or higher than 36 degrees were not favorable for Xylella to grow. But we wanted to know, is this temperature bacteriostatic or is bactericide? The bad news is that it's only bacteriostatic, so Xylella can recover not only at temperature below 4 degrees, up to minus 10 degrees and minus 20. It's not in this graph, but it's the last result. On the contrary, temperatures over 40, 42 degrees are lethal for the bacterium. So cold temperatures, the bacterium can recover. With the higher temperatures, Xylella cannot survive. So what happened in the tree? Because, I mean, we can reach easily 44 degrees, 45 degrees. So we did a study in which we monitored almond and olive trees with using specific probes inside the xylem vessels of the plant, in the trunk, at different depths, and also in the branches. So we saw that the minimum temperatures were coupled with the air, because it's much easier to record the temperature in the air. So we saw that almost the same temperature that was occurring in the trees was occurring in the air. Whereas the maximum temperature, the temperature were not coupled, so there were higher differences. And what we saw is that, for instance, in the silent tract, when the temperature is very high in summer, the temperature within the plant is 4 up to 8 degrees lower within the silent vessels. So Xylella could survive better summer inside the olive tree. And also we monitored what was happening in the roots, because Xylella can move from the tree canopy to the roots. So even if we are in cold regions, Xylella can survive winter, because as you know, the temperature in the root, the soil is a buffering platform. It's bad news for us, good news for the pathogen. So concerning the next tool for early detection for surveillance, the plant have different phenological changes, have different structural changes. So depending on the disease severity, different changes occur in the plant. Some of them can be easily seen by the eye, but as I told you, many of them are not detectable by the plant pathologist or by the farmer. So in collaboration with Pablo Zarco and Juan Antonio Navas, they lead this project in which we develop different remote sensing campaigns in Puglia, in olive, in Italy during different years, also in Spain in almonds, in Alicante and in the Balearic Islands. And also we went last year in summer to California to visit the almond trees affected by the bacterium. So what we do in the field is go and see 10 minutes? 5? Okay. See 3 by 3 what is the disease incidence, so what we can see by eye. We identify with a scale, you will show later. Then we take samples of 10% of the trees, and we do molecular analysis to see if that tree is particularly infected or not by the bacterium. So as I told you, we record the disease severity of each tree, and we also use proximal sensors to do different measures, the spectral signature of the leaves, the different stress index, water stress index, etc. With all the data, you can determine different indexes related to physiological, structural, and functional traits. And we do almost the same when we fly. So we use hyperspectral cameras, normal cameras, and thermal cameras. And when you identify if a particular tree is infected or not by the bacterium with the disease severity score that we have and all the spectral signature of the plant, we apply machine learning algorithm to see if we can differentiate before symptoms appearance the bacterium. Here you have some results of the proximal sensor. So you have results in olive, in almonds, and in grapes. So we can distinguish clearly plants that are healthy, not infected, for plants that are infected but have no symptoms. For instance, here, as you see, sorry, this is, for instance, this is with verticillium, but we have same result with Xylella, asymptomatic branches infected, and healthy branches. And with remote sensing, what we showed was that, for instance, the overall accuracy of the plant pathology in the field was 77%. So we have plants that were infected but not detected by the person in the field, whereas when we use remote sensing, many of the plants that were identified infected by remote sensing were indeed infected by the bacterium. So this technique shows important approaches. I'm not going to detail, but what we have seen is that we are able to distinguish Xylella infections from olive and almonds, and Xylella infections, for instance, from verticillium infections that are very similar in symptoms in olive, and also distinguish those symptoms from drought stress in olive. And just to finish, some molecular diagnostic tools. Probably you don't know, but this is the official procedure in each country. In Spain, the plant health authorities, the competence for doing the service is at regional level. So for instance, in Andalusia, we have the regional laboratory, and they have to use official tests that have been published in the legislation. Once a plant is positive, it has to be sent to the national reference labs in Valencia or in Lugo. And then for estimation of the subspecies, in the past was sent to our laboratory, and we have to determine the subspecies by molecular typing. So we also worked in developing very sensitive approaches that could detect the bacterium. This was a very nice study in which we sampled almond trees in Mallorca. So we went in using the dendrochronologic analysis, moving back to the year, because more or less you have an estimation of the age of the tree. And we were able to identify, using a nested molecular approach, that the plants infected in Mallorca in almonds were already, so the bacteria was already present in 1998. So, and the bacterium was identified in 2016 in Mallorca. So the bacteria have been there for many, many years. The almond trees were dying. They thought it was a problem of fungal diseases, and it was not the case. It was Xylella, the one that was killing the almond trees in Mallorca. So the EPO protocol for Xylella fastidiosa has all the molecular tests that can be used for detecting the bacterium. So what we did was compare all the official tests, and we saw that the sensitivity of the different tests was very different. So there were 3 of them that were very effective and were significantly more efficient than the visual assessment. And also that if we sample the gouty chip of the plant, is more sensitive than using the petiole that was recommended at that time for detecting the bacterium. You see the number of plants that escape if you sample petioles, as compared if you sample the gut of the branch. I'm not going to this because it's going complicated. And we are also developing a new tool that is based on target sequencing enrichment. Basically, what we have done is develop probes that are targeting Xylella sequence. So you have you use magnetic beads that sequence stick on Xylella DNA if it's present in the sample, in the plant, and then you use a magnetic to fish those sequences. So what you do is enrich your sample in Xylella reads. Why? Because if you extract DNA from a plant, 99.9% of the DNA is plant. Xylella is almost nothing. So if you do direct sequencing, at the end you don't get any reads. So using this approach and with a pipeline that we have developed, you see without capture, you have in green only plant DNA sequence, whereas if you use our approach in red, you have reads of Xylella. In that way, you are more efficient in identifying the subspecies. So in this way, we have identified that in Mallorca recently, the subspecies pauca that we had found there is identical to the one in Italy. So how this strain came from Italy or from Costa Rica to Mallorca, we don't know. But the severity of the olive trees affected by this strain is similar to that in Italy. So a big effort is being done to eradicate that particular outbreak, because if the bacteria move from Mallorca to the peninsula, you can imagine what can be the disaster. The take-home messages, I hope that you know now that Xylella is a priority pest, and why is the reason. That Europe has reacted well to fight against this pathogen. Indeed, it has changed the pest categorization rules and how the pathogens and the quarantine pathogens have to be analyzed now. That the only way to fight against this bacterium in research is through a collaborative project. Working alone, isolating, means nothing. It's a very difficult pathogen to work with, so in order to progress and move forward fast, you need to have alliance with other partners. And there is still hope, so we are working a lot in early detection of this bacteria by identifying what are the areas under higher risks in Europe. So if we know which are those areas, maybe we can use remote sensing approaches to flight or to go directly to the field and identify what are the host spots of plants that look like to have a profile compatible with Xylella infection, and then we can apply more efficiently the molecular tools that are very expensive, and you have to analyze hundreds and thousands of plants to find a positive plant. In that way, we can almost stop the spread and the establishment of the bacterium. And just to thank all the consortium, the BCIL consortium, the 4 PhD students that have developed a thesis on Xylella, and all the colleges around the world that have experience in Xylella and help the consortium to progress on research. And thank you for your attention.
Thank you, Blanca, for your nice presentation and very informative, and to be very restricted in the time. We have time for 1 or 2 questions.
Thank you, Blanca, for this nice, insightful presentation about the situation of a risk for our lifestyle, not only for the trees. So I have a quick question, two questions. One from the early detection. Do you think that this early detection approach for remote sensing can be implemented in the near future to satellites monitoring? This is the first. And second is if you know, and to your knowledge, if there is any response for antibiotics now. There is any antibiotics that can activate, can fight the Xylella infection, because actually the soil of the olive grove there is a lot of antibiotics. I don't know if this is a factor or not.
Mm-hmm. Okay. The first question, we have already done some analysis using satellites. The problem is the resolution of the satellites. Now there are new satellites that have hyperspectral cameras. The only thing you have to pay for the images, and you have to plan in advance what areas you want to fly. And we are doing research on that. Indeed, a few weeks ago, we have the resolution of the national projects, and we have one project on that. But we have already shown that it can work. The thing is that for intensive plantation, for instance, since the resolution is not high enough, maybe you can find spots, but there is some chance in the future to go on that way. It will be much easier than doing flights. And for the flights, we have already shown that it can be used, but of course, someone has to pay. The farmer cannot pay that. So this should be implemented by the regional governments or at national or regional level. And for the second one, there are some antibiotics effective against Xylella fastidiosa in vitro. I think some studies were done in the USA in Planta, but I mean, there is no chance because it's completely forbidden in Europe. And the use of antibiotics in plants is completely forbidden. And the way EFSA is going is decreasing at maximum the number of pesticides, of course, antibiotics. So for that reason, we are focusing in BCIL project in the use of biocontrol agents, antimicrobial peptides, natural plant extracts that can have some antimicrobial activity with the bacterium. We have already results in vitro, but that means nothing until you go to the plant and it's not phytotoxic for the plant. It can be applied in an easy way because how to reach the silent vessel of the plant and the entire canopy is still a challenge, but we will see. We need to continue working, so there is a bunch of things to do yet.
Okay, we have time for more questions. One more? No? Thank you very much, Blanca.
Okay, thank you very much. I want firstly to thank the organizing committee for this invitation, especially to Antonio. Thank you very much. My colleague, Dr. Julia, and me, we will talk about the antimicrobial strategies against phytopathogen in the olive sector and sustainability. This is the index we will use for this presentation. But first, I want to talk briefly about the olive crops crossroads. The olive sector is of immense importance. As you know, 750 million of olive trees are cultivated all over the world, being 95 to 97% located in the Mediterranean region. So this is the cultivated olives are considered as the primary element in the agriculture economy of the European southern country. Especially, in particular, Spain stands as the largest producer of olive trees and then the top producer of olive oil. Despite its vitality, the sector is facing several challenges related with the ecology and the economy. On the ecological front, this climate change and also the rise of different pathogens and also pests may have a great concern and a great impact on the olive production. So to deal with these challenges, this sector must adopt new and sustainable strategies to combat not only the pathogen, but also to adapt to a changing climate. Among these challenges, the microbial diseases are the major threat. They are caused by bacteria, fungi, and viruses. They are not only impact the olive production, but also they are involved in a broader range of issues, such as the pathogen spread, the economic costs, food security, migration, employment, ecology, and climate. So the pathogen that may infect the olive trees exceed 100. However, few of them are involved in serious economic damages. For example, the quarantine bacterium Xylella fastidiosa, as Blanca said before, which is considered as a global threat, but also other bacteria and fungi are considered, such as Pseudomonas and also the fungus Verticillium dahlia. So there are several strategies developed to deal with these challenges, with these microbial diseases. For example, the integrated pest management adopted or promoted by the Food and Agriculture Organization. This action includes the preventive control measures, the early detection, diagnostic and monitoring tools, and also the post-planting control measures. I'm going to focus on the use of biopesticide or biological control and the use of chemical pesticides, which are under some restriction. The chemical pesticide or the use of chemical pesticide is widespread because they are beneficial in the sense that they control the disease, they limit the pathogen spread, they guarantee the food security, and they reduce the waste. However, the research or different reports indicated the negative effect of chemical pesticide that exceed the positive effects. For example, they are involved in environment contamination, they increase the health problem, reduce the biodiversity, and also, and the most important, they increase the antimicrobial resistance, which is considered one of the major problems in the world related with the global health context. So, as an innovative strategy to deal with this pathogen and to reduce the use of chemical pesticide within the Green Deal, and also because biopesticide based on natural sources such as plants and microorganisms, they are most effective, they are considered as sustainable pest control system, commercially viable, and also safe. This biopesticide, now they are displacing the chemical pesticide. So this market also is facing some limitation because of the high cost and also the adaptation to changing environments. Within this innovative strategy, our research group is developing and working to develop a new biopesticide on the basis of the microorganism isolated from on the olive tree and also the surrounding environment. So in this case, we isolated a collection of bacteria from leaves, fruit, both raw and also throughout the fermentation process, and also from soil. And we found that these bacteria have antimicrobial activity against different phytopathogens and also animal and human pathogens. Why we choose the autochthonous microorganism bacteria in the olive orchards? Because the introduction of foreign microorganisms may disturb the biodiversity balance. So focusing on fruit and leaves, we isolated a collection of lactic acid bacteria identified at molecular level as Lactobacillus pentosus, and we showed both in vitro and in silico that they have a great probiotic potential. So from this collection, we selected the most robust, safe, and also attractive regarding their probiotic potential, some strands. One of them is CF6HA to analyze in depth. We sequenced the genome of this bacterium and we found that this bacterium harbored 1 chromosome and 6 plasmid with a plethora genetic information supporting not only growth, survival, and metabolism, but also defense and adaptation to different lifestyle, which is encouraging to be introduced in the olive orchards. Focusing on the antimicrobial activity, we analyzed in silico the presence of different the gene encoding different antimicrobial substances, such as the bacteriocin. We found the pediocin and also a plant-parasitic. Furthermore, regarding the secondary metabolite, which are also antimicrobials, we found the presence of gene coding for type 3 polyketide synthase. On the other hand, we also investigated the probiotic profile of this bacterium, and we found the gene coding for adenosine, exopolysaccharide, and also other genes, very interesting to promote their probiotic potential. To confirm the data obtained by in silico analysis about the antimicrobial substances, we screened this antimicrobial activity in vitro, both from the colony, from the cells directly, and also from the supernatant, because we need to confirm that these substances are released in the supernatant and are active against the phytopathogen affecting the olive crops. So we confirmed that both the colony and the cell-free supernatant were active against the phytopathogen of olive crops, such as the bacterium Pseudomonas syringae and also the fungus Verticillium dahlia. On the other hand, we also confirmed the activity against human and animal pathogens, such as Staphylococcus aureus, Bacillus cereus, E. coli, among others. So from this study, we conclude that this bacterium, selected on the basis of their robustness, safety, and the probiotic profile, may be considered as a talented bacteria with dual effect, active against phytopathogen affecting olive, tree, and other plant, but also active against other pathogen, animal or human pathogen, which encourage its use not only in the olive crops, but also in the food preservation. As biopesticide, the results are promising, but we have to test it or to apply in the field, so we require further studies. Just for your information, this bacterium was subjected to patent exam and now is under evaluation. And now I give the word to my colleague to continue exposing the innovative approach we are developing in our lab, but now against Xylella fastidiosa.
Okay. I'm a researcher at the University of Hain, and my work focuses mostly on the use of bacillus species as a source of biocontrol agents for major phytopathogens, and really the focus lies on on Xylella fastidiosa. So after Blanca's talk, I don't think I need to go into the specifics of Xylella again, but just to point out that, as we've heard before, Xylella fastidiosa full spread in the European Union could have a huge economic impact, as well as affect over 70% of EU's production value in olive trees, and could put over 300,000 jobs at risk. We've heard that, but I think it's important to remember that it's really a huge impact on the European Union. And so this is where we developed our projects on focusing on the development of biopesticides using bacterial spores. So this is a project funded first by a Marie Curie funding of the European Union, and now we're continuing this project with a European Regional Development Fund. And we want to use the antagonistic activity of bacillus strains for biopesticide formulations. Why do we focus on bacillus? So just for those of you who are not so aware of this species, this is a very interesting species because bacillus form endospores. These are dormant cellular structures, and that's actually the most resistant structures formed in nature, biological structures. And they have the characteristics of resisting high UV radiation, extended periods of drought or water scarcity, as well as the lack of nutrients. And this is, as we know, in times of climate change, these are one of the big challenges. So bacillus is a very interesting candidate. And in addition to that, bacillus species is known for producing a wide range of antimicrobial compounds. And in addition to that, bacillus is widely found in olive orchards. or in any kind of orchard, really, it's omnipresent. So how did we go about that? First of all, we established the culturable olive sporobiota in Spanish orchards. So that means the community of spore formers that are present in the Spanish olive orchards. So for this, we isolated spore-forming bacteria, and we did that in the soil surrounding the tree. But we also had a look at the leaves. So this is here. How does that work? So on the leaves, yeah. And here we had a look at the spore formers on the leaves, but also inside the leaves. So that's the endophytic spore formers that reside within the tree. And we have a community of 412 spore-forming isolates for now, which we characterized. We found a good antibiotic resistance profile, so that's important for safety concerns, but also for control issues. We also had a look at environmental tolerances, so that is just challenges that the bacteria might face in agriculture, so that is heavy metal tolerance as well as fertilizer treatment and so on. And they had a reasonable tolerance to these factors. And then we also found good antimicrobial activity against several pathogens. So first we had a look at the pathogens that are important in food production. So for example, Salmonella, Bacillus cereus, or Enterococcus faecalis. But then most importantly, out of this community of a bit over 400 spore-forming isolates, we had a look at the antagonistic activity against Xylella fastidiosa. And we tested 45 isolates. These 45 isolates were chosen due to their security profile. And 5 of these isolates did show antagonistic activity against Xylella fastidiosa and Verticillium dallei. And this was found both in the co-culturing assays, but also in the culture supernatant. So there is some kind of agent that is produced by the bacillus that has antagonistic activity against Xylella. And I just want to stress again what my colleague Hikmate already said. So the idea of using the bacillus species isolated from the olive orchards is to avoid introducing foreign agents into the olive orchards, but to use actually components that are already naturally present in the orchards. Yeah? So this is a sustainable strategy that does not alter the biodiversity of the entire orchard. And also in terms of regulatory hurdles, they are a bit lower there. So as I said, we found 5 of these isolates, and the next step was naturally to identify who are these candidates. So we did whole genome sequencing, and we found that the 5 isolates were Bacillus valesensis, so you see that in the first row here, Bacillus valesensis, Bacillus subtilis, Bacillus halotolerans, Peribacillus frigoritolerans, and Bacillus wittmanni. So these are the 5 candidates, but I want to highlight the 2 in the middle that have a lighter color, so the Bacillus subtilis one and the halotolerans ones, because these are the endophytic strains. So that means they already naturally co-inhabit the same environment as Xylella would do. Yeah, Xylella fastidiosa would do. So as I said, we did whole genome sequencing analysis to get information about these strains. We detected the size of the chromosome. There's no external plasmids available, and we also had a look at metabolic activity here. We were particularly interested in the polyketide biosynthesis, bacterial toxins, so that's also important, and antimicrobial resistance. Yeah, so that's important in terms of environmental safety. But then what we're really interested in is determining what is the agent that makes them active against the Xylella fastidiosa. Yeah? So in terms of future application. So we had a look at what genes are present and what we really expect. So you can see here the profiles of what kind of bacteriocins were detected in the different strains. So A, B, C, D is related to the different strains. And you can see that they're actually different. They don't really show the same profile. Yeah, so they all produce different substances, but with a similar effect, except for candidates B and C. So remember, that's the endophytic candidates, so they seem to have a similar mode of action here. In the next way, so this was the antimicrobial activity as by whole genome sequencing, but we also had a look at that in vitro, so by experiments, and we had a special focus also here on synergistic effects. Why synergistic effects? Because testing an environmental isolate in isolation actually doesn't really make sense, because the natural habitat of this bacterium is to live in a community. So it is only understandable that, or natural, that the activity the community would have against, for example, Xylella fastidiosa or other phytopathogens would be within the community. So are there any synergistic effects between these 5 isolates? We had a look, we tested then in our pathogens that I mentioned before, and surprisingly there was no synergistic effects for most of the combinations, but for the combination of the last 2 isolates, so that's the ones that are highlighted here in a circle. These are 2 of the isolates that come out of the same origin. Yeah, so these are the Malaga isolates. They did have a synergistic effect, and they first in isolation did not have any antimicrobial activity, but when you combine them together, then they start to be effective. So that was quite nice. 2 minutes. Oh. Good, I'll go quick. So one important thing that I would like to highlight is also the biofilm formation. That is a big issue in Xylella fastidiosa as well, as we've heard before. And we could detect, so just very quickly, we could detect a lot of biofilm forming genes by whole genome sequencing, and that also tied in very nicely with our experimental results, where we already saw by just culturing these bacteria, as you can see here, I don't know why that goes away. By just culturing these bacteria, you can actually see that the culture medium stays clear, but they form very, very strong pellicles on the air-water interface. So these are very strong biofilms on the air-water interface position, and they are so strong you can actually pick them up, and it's very difficult to mix. We then had a look at the biofilm strength, and all of the 5 isolates were identified as strong biofilm formers, and then strong biofilm formers. And again, for these Malaga strains that I mentioned before, we could detect synergistic effects. So if you culture them together, they form a much stronger biofilm than they would not. Why is this important? I repeat, the endophytic strains are the ones that are already in the leaves, or already in the conducive system of the tree. So this is the same natural habitat as Xylella fastidiosa would be. And then on the bottom, you just see visually how these biofilms look like. So on the left-hand side, you have one of the strong biofilm formers that really forms this extended sheets. And on the right side, you have a debris formation of one of the strains that is lighter biofilm former. And so I'm finishing now. I'm just going to finish now with the spore characterization, because as I said, our focus really is on the bacillus spores, which are extremely stable structures. They're very easy to produce, very cheap to produce, and highly resistant. So we wanted to have a look at how do they react with lysozyme treatment, though that tells you something about the integrity of the spore itself. We had a look in the second row at bleach treatment. Why is that important? That's disinfection strategies, because eventually you need to control these strains. And then also UV radiation. So UV radiation was good, could be better, but the UV resistance was also there. And then on the left-hand side, you have a visualization of this spore. They all look different, which is expected because they're all different species. But these were quite nice results, because especially for the lower 2, they have never been shown to have this outermost layer structure. So these ones here. And that actually enables them to interact very nicely with the soil and the plant. And then also, if you have a look at these arrows here, at the white arrows, they have these very long extended phyli. And these extended phyla itself also can have antimicrobial activity. So that is a very interesting point of research that is ongoing at the moment with us and with other research groups. So that was a short summary of these projects that I'm telling you. In conclusions, we have identified 5 olive sporobiter isolates naturally present in the Spanish olive orchards with antagonistic activity against Xylella fastidiosa. 2 of these isolates inhabit the same ecological niche. They show a strong biofilm formation and a good bacteriocin production, which might or might not be synergistic. And then they are environmentally safe. And we're still continuing the testing, but we already showed that they're environmentally safe. So both Hikmate and I, we thank you for your attention. That's our research group. The project was done, as I said, it's funded under the European Marie Curie projects as well as the European Regional Development Fund, and has been done in collaboration with the University of Cambridge. And we're very happy to talk to anybody who's interested.
Okay. Good afternoon. My name is Begoña Antón, and I would like to thank the organizers for the opportunity to present here at this international conference the work entitled Compatibility of olive endophytic biocontrol agents with beneficial fauna and entomopathogenic fungi. As you know, olive tree is a key crop for Iberian Peninsula, as well as throughout the Mediterranean region. But it is severely affected by complex diseases such as anthracnose that is caused by the species of the genus Coletotrichum. Also, olive knot that is caused by the bacteria Pseudomonas sebastianoi, Patovar sebastianoi. And these and other diseases can significantly induce low production quality and low production, and all quality. Traditionally, disease control of olive trees has been based on the use of chemical fungicides, which have certain limitations such as significant impact on the environments. For this reason, the main goal of the European Union is to create a sustainable food production system and to implement resilient agricultural practices with the aim to maintain ecosystems and increase productivity. In this regard, bioprotection through the use of biological control agents and other natural compounds are promising tools into an integrated pest management programs. One of the most promising sources of biological control agents could be within the plant itself. The plants have a complex microbiome that is composed by fungi, yeast, oomycetes, bacteria, and other microorganisms. They can inhabit the rhizosphere, phyllosphere, and endosphere, where endophytes are found. Likewise, the interactions between these microorganisms and the plant could be parasitic, neutral, or mutualistic. Among them, those endophytes that establish mutualistic relationships with plant are of particular interest, as they can promote plant health and provide protection against abiotic or biotic stresses such as pests and diseases. In olive, many endophytes are identified with antagonistic activity against several pathogens, as demonstrated by many works of our group and others. And it is the case of Arobasidium pullulans, a yeast that reduces sporulation and spore germination of Coletotrichum acutatum by 90% and 70% respectively in olive fruits inoculated with the pathogen and treated with the endophyte compared to the control. It also reduces disease incidence by up to 10-fold, and also disease severity by up to 35-fold, compared to fruits that were inoculated with the pathogen without the treatment. Okay. This other study evaluated the endophyte Penicillium commune, which induced the production of volatile organic compounds. These volatile organic compounds reduce the growth and sporulation of Coletotrichum nymphae, especially in olive fruits, suggesting that Penicillium commune had a greater effect against, on the fruit than on the leaves. There are other endophytes with inhibitory capacity against other pathogens, such as Pseudomonas sebastianoi, Patobar sebastianoi, which cause olive knot. And of 60 isolates evaluated from the olive tree and phytosphere, 28 significantly inhibit the growth of the pathogen. Okay. Especially Bacillus aeruginofaciens, strain P41, reduced disease severity by 44%, knot weight by 55%, and Pseudomonas sebastianoi population size by 27%. These and other endophytes have also a deterrent effect against in olfactometer assay against olive fruit fly or Bactrothera oleae, which is one of the main pests of olive, and also reduce the number of fly entries into olives in oviposition assays. Okay, all this background information suggests that these microorganisms, these endophytes, have a good potential, a biocontrol potential against two of the main olive diseases, and also against one of the two of the main pests of olive. But what about of the effects on non-target organisms such as beneficial fauna or entomopathogenic fungi? The study of this interaction and compatibility is key for safe integration into an integrated pest management programs. So, therefore, the objective of this study is to evaluate whether these 4 olive endophytes are compatible with Chrysoperla carnea larvae, a generalist predator, and Beauveria bassiana isolates. Okay, for the bioassays with Chrysoperla carnea, commercial larvae were individualized in petri dishes with filter paper, a food film with Festia eggs, and water gill spheres. 600 microliters of each BCA at a concentration of 10 to the 6 colony forming units per milliliter were added to its petri dish. Also, the insecticide EPIC was included as a positive control, and mortality was recorded daily, and individuals were isolated to confirm infection. As you can see here, treatment with the insecticide caused complete mortality at 7 days after inoculation. Treatments with Bacillus aeruginofaciens, Aurobasidium pullulans, and Penicillium commune did not cause any significant significant effect on the larvae. And however, Alkaligenis faecalis showed some negative effect, reducing survival rate by 22% on day 7 compared to the control. Data were analyzed until a control larva died on day 7. However, the experiment continued for 25 days to ensure that that many of the larvae pupate and reach to the adult stage. And for the dual culture assay, 2 Beauveria bassiana isolates were tested on mixed culture medium with PDA and LBA, confronting them with each BCA. For these 2 paper discs were placed 5 centimeters apart. One were inoculated with 10 microliters of each isolate of Beauveria bassiana, and the other paper disc were inoculated with its BCA. Also, Beauveria bassiana was inoculated 4 days beforehand to compensate for its lower growth rate. The plates were incubated for 8 days at 28 degrees, and the mycelial growth was measured every 2 days. And percentage of inhibition were calculated to each treatment. As you can see in these graphs, only Bacillus amelioicoephathians and Penicillium commune significantly inhibit the mycelial growth of the isolate AC93GF, although in no case exceed 12% compared to the control. With the isolate A67, Penicillium commune showed the greatest inhibition, but although in no case was more than 60%. It is noteworthy that, as you can see here, Arobacillium pullulans did not present a significant inhibitory effect against Beauveria bassiana, against any of the isolates, as they continue to grow together. However, a weaker Beauveria bassiana mycelium was observed in the overlap zone. Furthermore, in some cases, it was observed that BCAs were inhibited by Beauveria bassiana. This is a fact that we must study, that we must further study, as slight competition could be mutual. And to conclude, the endophytic species test showed potential into IPM programs against olive tree pests due to their biocontrol effectiveness and limited harmful effect on beneficial organisms. It is important to evaluate compatibility at different stages of development, especially in species such as Chrysoperla carnea. Further testing with other non-target beneficial organisms is recommended to confirm the absence of adverse effects, and validation of these results under field conditions will be essential for their practical applications. And thank you all for your attention.
Begoña, for showing us the potential of endophytes in biological control agents. And the next communication will be made by Sofía Silva from the Polytechnic Institute of Bragança.
So, good afternoon. I would like to start by thanking the organization for selecting this work. My name is Sofia, and I will present our work on endophytic shifts across olive tree lifespans and their potential role in stress resilience. This study is one part of my PhD project. I will start with a small introduction about the work and their importance. So, heat waves and droughts are becoming a more frequent phenomena, causing losses in agriculture. This climate change also have consequences for olive production. Even though olive trees are drought tolerant, water stress reduces flower and fruit production, decrease leaf area, and limit photosynthesis. We all know that olive trees are extremely important crop in the Mediterranean region. In Portugal, the Trás-os-Montes region is the second largest producer of olive oil, characterized by traditional olive groves with centenary species, and they grow in high temperatures and under drought conditions. It has been described in the literature that with age, crops tend to be more resistant to various extreme factors. And this is interesting because it's very common to observe that older trees are more resistant to drought, but the mechanisms behind this resilience are not yet fully understood. But one possible explanation for this is the association with endophytic microorganisms. So endophytes are fungi and bacteria that lives inside plant tissues without causing disease. And they have a lot of benefits. For example, they can promote plant growth and protect host plant against pests and diseases. They also improve tolerance to biotic stress and abiotic stress like drought, salinity, or extreme temperatures. These endophytes may be key players in the resilience observed in older olive trees, but what remains unknown is the relationship between the plant age, the endophytic microorganisms, and drought resilience. So, our question is if do endophytes change with the age, and if they do, this can explain the resilience of older trees. So, like I said, because the socio-economic importance of this crop, it's important to understand the mechanisms that confer tolerance to drought stress in these plants. So, our main objective is to develop strategies to increase your resilience to climate change. In this presentation, I will focus in one part, analyzing using cultivative methods if the endophytic community of leaves and roots varies with the age of the trees. To do this, we select olive trees from the same cultivar, but with different age. Less than 10 years, which we call the new ones, VN, around 50 years, which we call the middle ones, VM, and more than 100 years, which we call very old trees, VV. We collect samples from leaves and roots, and then we sterilize them on the surface, and isolate both fungi and bacteria using culture-dependent methods. After that, the isolates were identified by sequencing the 16S for bacteria and the ITS for fungi. The statistical analyses were performed by using some bioinformatic programs like CAP, Pest, and RStudio. Moving on to the results and starting by the diversity analysis. Here, we can see that bacterial diversity in leaves decrease with tree age. In contrast, fungal diversity in leaves increase with tree age. But in the roots, we don't see any significant difference in bacterial diversity. In contrast, fungal diversity in roots increase with tree age. So, overall, fungi diversity tend to increase with tree age, but while bacteria remain stable or decrease, especially in leaves. Moving on to the relative abundance. In leaves with age, the abundance of Pseudomonas increase. However, the abundance of Bacillus decrease with age. In contrast, in fungi, with age, the abundance of Alternaria and Fusarium increase. However, the abundance of Phaeosphaeria decrease with age. In roots, we don't see an increase or decrease in the abundance of bacteria. But in fungi, with age, the abundance of Dactylonectra increase. And some genera, like Trichoderma and Phomopsis, tend to appear with age. On the other hand, the abundance of Chaetomium, Clonostasis, and Fusarium decrease with age. The taxonomic composition also reflects these patterns, with different dominant genera in leaves versus roots, and in young versus older trees. For example, in leaves, Bacillus and Mycobacterium are more positively correlated with the youngest trees, Orobactrum with the middle ones, and Pseudomonas with the oldest ones. In fungi, we see the same pattern. We have some genera that are more positively correlated with the age of the tree. In roots, we see the same, Bacillus and Lisinobacillus correlated with the news, Pseudomonas with middle ones, and Nealia and Prestia with the oldest ones. The fungi, the same, Chaetomia with the news, Mortierella with the middles, Trichoderma and the other two genera with the oldest ones. And all of these genera have different ecological functions. Some of them can promote plant growth and helps in stress tolerance, like Bacillus, Pseudomonas, and Trichoderma. Other species other species as pathogen species like Alternaria, Diaport, and Fusarium. So, this functional diversity means that the change associated with age can both increase beneficial interactions and increase potential risks. To conclude, tree age strongly influenced the composition of endophytic communities with functionally relevant shifts. In leaves, bacterial diversity decrease, but fungal diversities increase. In roots, bacterial diversity is stable, while fungal diversity also increase. Older trees harbor both beneficial genera, like Pseudomonas and Trichoderma, and potentially pathogenic ones, like Diaporte and Fusarium. This suggests that age-related resistance may result not only from plant physiology, but also from the dynamics of endophytic communities. In a future work, we will explore this in more detail, using culture-independent methods and experimental inoculations under drug stress. This will help to clarify if it is plant age, endophytes, or both that confer resilience. Thank you.
To keep for watching my presentation. So we're going to talk about nematodes, but plant-parasitic nematodes. But I don't want to keep the message of nematodes are bad. Nematodes could be they are on three stages and trophic levels. Plant feeders are these ones. So they keep the light from plants and keep on the system on the soil. So they do important functions in the soil, nematodes in general. About 10–20% of nematodes in natural ecosystems are plant parasites. But population imbalances is what cause a disease. In here you see two examples of important population balances. You see that it's the same species is the majority of the community, so that make disease. So promoting soil health, like increasing organic matter, reduction of disturbances, crop rotation, usually reduce problems with plant-parasitic nematodes. Olive is one of the important crops in the Mediterranean economy, and the olive tree rhizosphere is home from both free-living and plant-parasitic nematodes. Up to date, it's around 250 plant-parasitic nematode species are described in inhabiting olive grove rhizosphere. That means that not all of them cause disease, but they could live on the roots on the olive, and also they could feed alternatively on olive or also specifically on weeds. It's estimated that 10% of the productions of many crops are caused by plant-parasitic nematodes. But from these 250, there are groups that are important plant-parasitic nematodes, like Meloidogaine species that are root non-nematodes, Pratylencus root lesion nematodes, cyst nematodes, some species of Heterodera, spiral nematodes, Helicotylencus genus, these are endoparasites, and some as Criconemoides are ring nematodes that are ectoparasites. Just to understand how nematodes damage the trees, the damage of nematodes is highly related with the number of them in the soil. So if you see this graph, is production and nematode soil density. So as we increase nematode soil density of one species in the soil, the production is reduced. And there are two levels, like tolerance limit, that is when statistically we found a reduction in production or in growth, and an economic threshold, that is when the measure to control the nematode is economically rentable as we increase the production of the measure of control. Specifically in olive, it's not much studied the damage of this threshold, but if you see at the bottom, Meloidogaine species, depending of the species, 10 juveniles per 100 grams of soil could cause damage, reduction of growth in plants. It's also important to know that the olive is a vigorous tree. It's not like an horticultural crop. So in woody species, the damage is mainly caused by seedlings. Then we have a high population of nematodes in soil. In bending, then we put another line of tree in a white row trees. And in a stress situation, for example, drought or damage on the roots. So the objective of this talk is, in an agricultural environment, how are plant-parasitic nematodes affected by environmental factors, and which fungal biocontrol agents are dealing with them and their requirements to biocontrol. So main of the data is inside the project Soil Alive. So we sample in 5 countries, Portugal, Spain, Morocco, Italy, and Greece. We study 294 samples in 52 fields, 20 organic and 32 non-organic. In order to identify the nematodes, we use integrative taxonomy, because nematodes, they have a similar morphology, but they could be different species. So integrative taxonomy is a method that combines morphology, morphometry, and molecular markers. Because in nematodes, it's common the presence of cryptic species. So it's the same morphology, but in genome and in ecology are different. So in this, for example, we found a high diversity of Paratylenchus species, Paratylenchus species. Also, in order to analyze this data, we have 71 variables analyzed: climate, spatial, management, soil, pesticide, like residues, and plant-parasitic nematodes. So we found a great diversity of plant-parasitic nematodes in all Mediterranean countries. Species like Syphinema, that are these big ones, big nematodes with a long stylet, Helicotylenchus, Rotylenchus, Paratylenchus, Criconemoides. And the inventory of species is 111 species belonging to 32 genera. The most diverse groups are 16 species of Paratylenchus, Syphinema species 16 species, and Helicotylenchus 7 species. We also describe in this project 3 new species, 2 Longidorus and 1 Neodata in collaboration with colleagues with Morocco and Spain. So if we go for, this is the analysis of the genera, the importance of the genera we found and how important are they in numbers. But if we are studying the most pathogenic that could cause disease, so we found genera, the root-knot nematodes. So we have a prevalence of around 50%. Paratylenchus, that is root-damaging species, so it's around 35% prevalence in soil. Helicotylenchus, 92%, Rotylenchus, 25% of the samples, and Paratylenchus is around 60%. So it's important to point that some groups are highly prevalent in all the samples, so they could be potential damage. And in the case of Helicotylenchus, we have samples with 20,000 nematodes per half a liter of soil. So they could cause a potential damage in replantation or if we change the crop. If we study in this case, we have the amount of free-living nematodes that are all the species, all the nematodes that are not plant-parasitic nematodes and plant-parasitic nematodes. We only found differences among countries. So Greece and Morocco has the highest densities in free-living nematodes, while in plant-parasitic nematodes we have in Greece and in Morocco and in Spain. And the interaction in management, we didn't find any between organic and non-organic. We don't find difference in free livings or neither in plant-parasitic nematodes, and the interaction was not significant. If we go to this group, then it's a highly important plant-parasitic nematodes, the root-knot nematodes. They produce these knots on the roots, and they become sedentary, so they interact in the absorption of water. We found 4 species, Meloidogaine javanica, Arenaria incognita, and hispanica. The prevalence was around 50% of the fields. Incognita in 3 countries, hispanica in 2 countries, javanica in 2 countries, and Arenaria only in Morocco. And the highest level in root, so it signifies that we have a potential damage, is 24x per gram of root in Spain. Meloidogaine javanica is the most damaging nematode in olive of this group. Comparing to our previous sampling in Andalusia, in wild olive and cultivated olive with a good amount of samples, 123 and 376. So the prevalence is lower in Andalusia, but we found a higher diversity of species. We found Javanica incognita, Arenaria, Apla, Betica, and a new species that won't describe it there, Oleae. But we found a high prevalence of Meloidogaine javanica, so it has a potential to be damaging in plantations. I point here that Meloidogaine javanica, even at 0.1 juveniles per centimeter cubic of soil, could cause a reduction of around 40% of the growth in pot experiments in olive. Another genus that is surprising that we found some samples with 20,000 nematodes in soil is Helicotylencus. They can adopt an endoparasitic position. They put the anterior part inside the root, as you see here, and they feed through the stylet. So it described a reduction of 75% of the growth with 1,000 Helicotylencus digistera in pot experiments. And we found from samples from field, from plantations in Spain and in Cáceres, that Helicotylencus microlobus at 40 nematodes per centimeter cubic of soil, they could reduce the company, the farm sent us the samples, and the seedlings, they didn't grow. But the pathogenicity in olive trees has not been clearly studied. And a scientific question that we are going to work in the future is why specific growths, because these are only specific growths, how they reach so high densities in soil and root. So if we take the main sampling from the project, we studied the richness, the beta diversity, and the global dissimilarity, and we didn't find a specific pattern in Europe. So we are going to study if there is some pattern, studying with the ecological, the 71 variables that we measure ecologically. So we do a variation partitioning. This part was done by my PhD student, Rosana Salazar. And we that an environment explained around 50% of the variation, and explained 82% of the relative variation. And climate, soil management, and residues explain the majority of the. It passed automatically. So in this case, what happens here? So we found the stochasticity is low compared to other studies, probably because of the geographical scale. If we study the percentage of variation, the climate and soil are the major explanation. And we found a great integration between climate and soil. So we thought that the management of the covers and vegetation could also change the management of the nematodes. So we test this hypothesis. So my student started to study this in a structural equation modeling. So we study climate, organic, non-organic, residues, pH, chemistry, physics, texture, spatial, and management, and what are the direct and indirect relationship between the system and plant-parasitic nematodes. So we found that some variables are quite important, but like management, but the most important parameters is climate in our sampling. So our question was, how does increasing aridity affect plant-parasitic nematodes? So we found that we have a dissimilarity of the community decrease, as you see in the graph, and it's found that the environmental disturbance causes homogenization because of drought. So we found that in plant-parasitic nematodes, they found is reduced the dissimilarity and the evenness, but we don't see a reduction of species when the climate, when the area sampled is more arid, have less water. So we found a homogenization, reducing dissimilarity and evenness. And how it affects, which trophic group it benefit, this reduction of beta diversity. So if we plot the ratio plant-parasitic nematode in respect to the total, including free livings, we found that it's related to an increase in the proportion of plant-parasitic nematodes. So then we study the second question, can different management strategies cushion the effect of the certification on plant-parasitic nematodes? And we found that we didn't find any significant difference between treatment, between organic and non-organic in our sampling. But we found that in organic, the dissimilarity is reduced, but the richness is similar, a reduction in the evenness. In organic also reduce the dissimilarity, but we have a little bit more of a species of always talking about plant-parasitic nematodes, and it's reduced moderately the evenness, as you see in the graphs. We now go, are the nematodes, so we now go to the nematophagous fungi. So these are potential biocontrol agents to control plant-parasitic nematodes and nematodes in general. So we do 2 bioassays with the soil. We do a qPCR detection on nematode community. So we extract the nematode community and we study the DNA, and we do a specific qPCR. And we also do a bioassay. This is egg bite bioassay, and we have 2 lines of soil. We put a bite of eggs, so fungi growth, and if they attack the eggs, we count how is the activity in the soil. So from the first one, we study different biocontrol agents. You see here the species and the mode of action. Some of them are nematode trapping fungi. You see here how they trap meloid organic juveniles. They make a ring and they attract the nematodes. And other are endoparasites, that they attack the nematode, and other are parasitic fungi, as you see here. So we found the nematode. So we have a different set of nematode activity. So the question where organic field management influenced the activity of these nematophago fungi, and which environmental and management factor influenced the activity of this. So with the qPCR detection on the nematode community, we found 1 species that was highly prevalent, that was Purpurosilum lyrasinum. We found this in all the countries. And the other species were not so abundant, and it depends on the country. If we study the same species, but in organic or non-organic, we don't found any pattern. We don't found that in organic management we have an increase of the presence of these nematodes, because the ratio is similar, 70, 80. If we go to the biocontrol egg bioassay, we study all the parameters similar to the nematodes, but the most important influential effect, like in nematodes, in our study, is the aridity index. So as we have more water, we have more biocontrol. So in this case, the nematophagous fungi activity is closely related to humidity. So we have here the infection rate is reduced as the sampling point is drier. It's in a drier climate. And we didn't find any difference in the management. The highest activity biocontrol was in Greece, even with some samples of infecting more than 10% of the eggs in the bioassay. But we also found the high presence of Trichoderma marcianum, then it's already a described biocontrol agent for this species. We also, the main genera we found in this bioassay was Trichoderma and Fusarium. Also are the most abundant in nematodes. We found a difference in the country. So Portugal, and here it's Portugal and Greece has the more humidity. You see the precipitation evaporation. Portugal and Greece are the highest, and was the highest activity. But no any significant relationship between organic and non-organic inside the country. So the future of our analysis will be a study not only taxonomic diversity, but about functional diversity, because we found that if plant-parasitic nematodes are endoparasitic, ectoparasitic, or semi-endoparasitic, could have some incidence on the explanation in the variables. What happens when comparing agricultural and natural environment? So we are working with this with our colleague on the project. And we need to study other relationship yet go deeper on the modeling. So I thanks the Soil Olive project and all partners that help in the sampling, and also the people in the team that work with the analysis. And here is Rosanna that is doing the PhD. So I think I am on time. Many thanks.